Hello everyone, welcome to the screencast on the human immune system. Recall that the immune system is a system that is designed to protect us against infection, and it does that by identifying and killing pathogens and, yes, even cancer cells. So we're going to take a look at the first half of the immune system here in this screencast. The immune system is divided into two major divisions. The first of these divisions are what we call the innate defenses. Innate means born with. So these are the defenses that are, so to say, online as you're even born or even before you're born. The innate defenses represent the primary line of defense against invading organisms. It is, however, a very non-specific line of defense, meaning that it works generally against all pathogens. The innate defenses are mobilized immediately then upon infection. We don't have to wait around for a period of time in order for the innate defenses to take effect or have an effect on pathogens. The innate defenses are divided themselves into two subdivisions. These make up the first, what we would call, two lines of defense of the immune system. These first two lines are referred to as barriers to entry and the phagocytic white blood cells. And in just a few minutes, we're going to be taking a look at each of them. The next line of defense, or the next division, I should say, major division of the immune system is the acquired defenses. The acquired defenses represent a third and specific line of defense. Specific means that this line of defense is designer in nature. It is designed to take care of specific pathogens. And when this line of defense is engaged, it can only work against that specific pathogen, no others. The acquired defenses do take time to react to the invading organism, so they are not online immediately. It takes time for them to develop because they are specific. And these acquired defenses, as we said, do represent a third line of defense. All right, let's take a look at each of these lines of defense more specifically, starting with the innate defenses and that first line of defense, the barriers to entry. Your skin is your first and most important barrier to entry. The skin cannot be penetrated by most organisms. However, the skin can be damaged, which can allow organisms to penetrate. And of course, there are natural openings in the skin through which organisms could enter the tissues of the body. So while it is the most important barrier, and it's a very effective barrier, pathogens cannot get by it, there are these natural openings or potential for openings that pathogens can get by. So the skin needs some supporting characters, some supporting structures. The first structure we take a look at, in addition to the skin, is the mucous membranes. Recall that mucous membranes you learned about in AP1 are wet membranes that line body cavities and surfaces that are exposed to the outside. So we're going to see mucous membranes lining things like the respiratory tract, the digestive tract, the reproductive tract, and the urinary tracts. These all are exposed to the outside with the respiratory tract and digestive tract via the mouth, the reproductive and urinary tracts via the urogenital system. These mucous membranes produce a sticky protein substance called mucus, and that mucus has the ability to trap many different microorganisms that make it past the initial barrier to entry. In addition to the mucous membranes, there are also certain chemicals that can aid the skin in helping prevent the entry of pathogens into the tissues of the body. Some of these chemicals include things like sebum. Sebum contains lactic acid and fatty acids that inhibit the growth of some bacteria and fungi. Recall that sebum is produced as an oily substance that's produced by the sebaceous glands of the body. And because of the presence of lactic acid and fatty acids, that's going to help to lower the pH of the surface of the skin, thus making it more difficult for pathogens to get past. In addition to sebum, There are other chemicals like perspiration, saliva, and even our tears. Remember, all three of these contain the antibacterial enzyme lysozyme. Uh, Saliva, by the way, also washes microbes off the teeth and tongue, so it's very important. People with problems with dry mouth can have problems with halitosis, bad breath, problems with their gums, because without the saliva to continually wash the microbes off the teeth and tongue, these microbes hang around and can cause bad breath, cracked gums, and other problems. Tears also, by the way, do wash the eyes. We talked about that in our look at the special senses. In addition, we also have acidity that we can take a look at. And for example, hydrochloric acid is secreted by the mucosa or the inner lining of the stomach. This inhibits or kills many types of bacteria. Most bacteria cannot live at low pHs. For example, the internal cavity lumen of the stomach, then that pH is somewhere down around 2 with the hydrochloric acid. Most bacteria cannot live there. And there is also acidity associated, for example, with the vagina. The acidity and its thick walls also helps to discourage the presence of pathogen in these areas. And we also take a look at urine. Urine is important because not only does it help us to excrete waste products, but urine also helps to flush bacteria from the urinary tract, helping to prevent urinary tract infections. Next up, we can also take a look at another aid to the skin, which is the resident bacteria, or what we call normal flora. So there are good bacteria that actually live in various niches in our body, for example, the mouth, the intestines, and other areas. These resident bacteria fill almost all available ecological niches in the body, and they produce various chemicals which work to destroy other bacteria that might compete with them for that niche in the body. Remember, these bacteria are living inside our body, and they're getting the four main things that they need for survival. They're getting warmth, they're getting moisture, they're getting darkness, and they're getting nutrition. They have a pretty good setup there, and they want to live in that area, so they produce chemicals that might discourage pathogenic microbes from entering and possibly taking over that same niche. Next up, we'll take a look at the second line of defense. And this is also part of the innate defenses. And this second line of defense is going to be best represented by the inflammatory response. The inflammatory response is going to employ mainly neutrophils and macrophages. And these neutrophils and macrophages are going to perform phagocytic functions. They're going to surround, engulf, and destroy pathogens. There are four hallmark symptoms of the inflammatory response. That inflammation, this second line of defense, has been engaged. These four hallmark symptoms include redness, heat, swelling, and pain. And we'll take a look at why each of these symptoms occur with the inflammatory response. So the inflammatory response we can divide into several steps that enable us to understand it a little bit better. Now, I'm going to divide it into three main steps, but you might hear others talk about 4 steps or 7 steps or more. As I take a look at it, though, I'm going to divide it down into the essential steps. You'll see what I mean by how we could add steps to it as we move along here. But the first step of the inflammatory response would be the release of chemical mediators. So the pathogen makes it by the initial barriers to entry, and now the pathogen enters the tissues of the body. Tissues become damaged. These damaged tissues and mast cells associated with the tissues, often found in the tissues, will release chemicals such as histamine, that might cause capillaries to begin to dilate. This is going to increase blood flow into the area and cause the first of the hallmark symptoms we talked about, the redness that we see because of the great increase in blood flow. This is going to result in, with the increase in blood flow, also a movement of neutrophils and macrophages into the area. And we can focus primarily on the neutrophils. Now, here's where I have step 2, and I call step 2 diapedesis. But some individuals may put a second step in between R1 and 2 here and call that margination. So what happens in margination is once the chemical mediators are released and the blood vessels dilate and blood flow increases into the area, neutrophils move into the area. And initially, the neutrophils marginate along the inside of the blood vessels. That is to say, they line up along the blood vessel walls until the blood vessel dilates enough to create enough gapping between the cells of the endothelium that the neutrophils can slip between those gaps now and move out into the tissues. And once they move out of the bloodstream and into the tissues, the neutrophils can begin to phagocytize any debris, dead cells, or in the case of the immune system, bacteria or other microbes. Well, recall that the neutrophils are not much bigger. We talked about this when we were going through the blood, that the neutrophils themselves are not much bigger than the microbes that they may be removing. And so, when those microbes are destroyed, the neutrophils are destroyed as well. This results in an accumulation of pus, or the development of purulence. Purulence or pus is dead neutrophils that accumulate in the area. So an accumulation of pus or neutrophils is a sign that infection is going on. The next step of the inflammatory response then is the release of cytokines. The neutrophils, as they're dying, begin to release certain chemicals. These chemicals will attract monocytes into the area, kind of a backup system, and these monocytes, whenever they get into the area, they will fuse together to form large phagocytic cells called macrophages. The macrophages themselves then will enlist the help of T cells and B cells, and those T cells and B cells will be engaged in the third and final stage of the immune response. And we'll be taking a look at T cells and B cells a little bit later on. Okay, so can you see why, before we go on to the complement system, can you see why the 4 hallmark signs of inflammation occur? So histamines are going to be released. That's going to dilate blood vessels. When those blood vessels dilate, that's going to increase blood flow into the area. We're going to have redness. There's going to be inflammation because with that increased blood flow and the gapping, there's going to be more fluid exchange into the tissues. The fluid's going to accumulate into the tissues, and there's going to be less fluid recovered from the tissues during capillary exchange. There's going to be inflammation. There's going to be redness from the increased blood flow, and there's going to be pain because the inflammation will put pressure on pain receptors in the tissues in those areas. All right, moving on. The next component of the innate defenses are what we refer to as the complement system. The complement system, well, I have here, the complement system consists of 26 blood proteins that work together with the immune system to help destroy bacteria and viruses. I have 26. I've seen as low as 11 blood proteins that different authors talk about, and I've seen as high as 30, so I just kind of picked an intermediate number. You'll see a different number a little bit later on. No matter whether we talk about 11 or 26 or 30 proteins that work together to destroy bacteria and viruses, these complement proteins all work together to do things like attack and break down the cell walls of microbes. Let's say, for example, bacteria. That's where we'd find cell walls. attract phagocytes into the area. So histamine could be considered one of these proteins in the complement system, and also to stimulate inflammation. In addition to these 26 blood proteins, there are also other proteins referred to as interferons. Interferons are proteins that work against viruses. These are proteins that are produced by cells that are infected by viruses. These interferons will bind to receptors on target cells near the virus-infected cells, and that receptor binding then will stimulate those target cells to produce substances that interfere, thus the name interferon, with that viral replication. Interferons are used in the treatment of viral infections such as hepatitis C and, for that matter, HIV or AIDS as well. Now here's a screen that shows a kind of a diagram of generally the way that interferons work. So let's kind of take a look at this diagram. And here on the top left of the diagram, we have a cell, let's say, for example, that has been infected by a virus. We can see the virus here at the top of the cell attaches itself, opens up a hole in the plasma membrane of the cell, and then the virus inserts its genetic material into the cell. Once the genetic material is inserted into the cell, the genetic material then will move to the cell nucleus and it'll connect itself into the genetic material of that cell. Once it has become connected to the genetic material of the cell, the viral nucleic acid will be replicated, it will be transcribed, and then translated by ribosomes, and the net result will be that the virus will be replicated inside the cell. So basically what happens is this cell now gets taken over by the virus, and it does nothing except for to make new viruses. That's ultimately going to result in the death of that cell. It's not going to be able to take care of itself anymore. But prior to being taken over by the virus, the recognition of the virus nucleic acid is there. There is an interferon gene already present in the genetic material of the cell. That interferon gene becomes activated and it produces a transcript of messenger RNA that is read by ribosomes, and ribosomes begin to synthesize interferon. So here we can see the interferon that's been synthesized, and that cell now releases the interferon into the surrounding environment. The interferon then itself will bind to the surface of neighboring cells. So these cells on their plasma membrane, remember, will have receptors. These receptors, whenever they are bound with interferon, will send a signal to the healthy cell. Remember, this is an uninfected cell now, to begin, before it is infected, to manufacture antiviral proteins. So here on the cell is an antiviral protein, or an antiviral protein gene, I guess I should say. And that gene will become activated. It'll be transcribed and translated then by ribosomes to make antiviral protein. So that if this cell then becomes infected by a virus, as we can see in this top right diagram, when the virus inserts its DNA, it'll have no effect on the cell because the antiviral proteins will block the viral replication by blocking the ability of the viral nucleic acid to insert itself into the genetic material of the healthy cell. So while this cell, it's too late for it, it secretes interferons that help other cells protect themselves against infection. That's how interferon works, generally speaking. Okay, so here is just a diagram, a inset that I put in here just kind of reviews everything as far as the first and second lines of defense are concerned. The innate defenses. So we would have physical barriers like the skin and secretions of skin like sebum. We would have phagocytic cells, which remove debris and pathogens. And so we can see some of the various phagocytic cells. You may recall some of these different names here: neutrophils, macrophages, eosinophils, and monocytes. There's also extracellular killing, which we haven't gone over yet, but we will in the future. And we have the inflammatory response, which we talked about. I gave you 3 steps, showed you a possibility for 4 steps. Here in this diagram, 7 steps. Again, all depends on who you're talking to. Fever is another innate defense. Fever mobilizes defenses, accelerates repairs, and inhibits pathogens by making it more difficult for pathogens to reproduce. As a result of fever, that is when the body temperature rises above 37 degrees Celsius, pathogens have more trouble reproducing because their proteins have trouble folding up because of the higher temperatures. That gives the body's immune system a chance to defeat the microbes before they can grow into significant numbers. Here we can see interferons, and we've talked about those, and of course the complement system. All right, that's going to take us to acquired defenses, and we'll talk about acquired defenses next time.